Okay, I will then start with my presentation. So this virtual educational training, as I said, I'm Laura Suter-Dick. I will be moderating this, and it consists roughly of 3 parts. I will introduce the topic of the 3-dimensional liver models with a PowerPoint presentation. I expect to take about 20 minutes to do this, so that you know what we're talking about and what you're going to see. The second part is recording, a video recording of all the practical steps in the lab. So we have pre-recorded this because otherwise we would need like 3 days to show all the steps that we need to do. So this has been pre-recorded for you, about 25 minutes. And after that, of course, we will be open for questions and answers. And the title, as you know, is the 3D HepaRG model, microtissue generation and applications. So the objectives of this whole educational training, virtual educational training, and including this session, is of course to introduce you to some in vitro approaches, promoting alternatives to animal experimentation. And as you know, the 4 partners are the FHNW, or the University of Applied Sciences, Northwestern Switzerland, the Faculty of Science in the Lebanese University, where I believe all the students are coming from, BioPredict International, and Edelweiss Connect. This 3rd session, the first 2 sessions, you have seen the cell culture basics, and now we're going to go to a more advanced model, how we can use the HepaRGs in a more complex microtissue generation. To myself, since you don't know me, of course, I'm from a different university. I work in vitro toxicology and cell biology at the University of Applied Sciences in Northwestern Switzerland. This is in Muttenz, near Basel, if you know Switzerland. I worked for a long time in pharmaceutical drug development at Hoffmann La Roche, a pharmaceutical company here in Basel. And my research focus is mainly on advanced in vitro models. I'm very interested in reducing, replacing, and refining animal experimentation. As a toxicologist, I'm heavily involved in toxicological applications of these in vitro models. And of course, we're in a university, so we teach to the bachelor and master's degree, and we also supervise some PhD students. The school generally is here in Muttenz. It has several areas. One of them is life sciences, and the only thing I wanted to highlight is that we work very closely with industry, and that's why also we're having this training also in collaboration with BioPredict and Edelweiss Connect. So now to the topic. The liver cells. And I know you heard a little bit about the liver cells, in particular about the HepaRG, definitely in your biology courses, but also in the previous training. I just want to show this this depiction, with this figure depicts the different liver cells. In the center, the parenchymal cells. The biliary epithelial cells or cholangiocytes, and these cells can be generated from the HepaRG. The HepaRG is normally differentiated into hepatocytes. They can also be differentiated into cholangiocytes. That's very important in the liver for the liver function and the responses to toxicants, for example. In disease. Microphage is important for the immune response in the liver. Another one is the hepatic stellate cell. The hepatic stellate is only about 5% of the total cells in the liver, but the hepatic stellate cell, however, has a very important function in tissue repair and in collagen extracellular matrix deposition. In addition, we have the liver sinusoid which cover the sinusoids. The model we're gonna be showing you today focuses on these three different cell types. We will focus on the parenchymal cells, namely the hepatocytes, and we will co-culture these hepatocytes with macrophages, or cells which represent the Kupffer cells, and cells which represent the hepatic stellate cells. So therefore, we're going to speak about the three-dimensional system, but also a co-culture system with three different cell types. And you will see how this is performed briefly. So here in the form of a table, we're going to represent the hepatocytes with the HepaRG cells. You know a lot about them from BioPredict. We're going to represent the Kupffer cells, these resident immune cells, by THP1. This is a human monocyte-derived cell line, which fulfills many of the functions of the Kupffer cells. And we will represent the stellate cell by an immortalized stellate cell line that has been provided by Bernd Schnabel. So three different cell types will be used in this model. To note is that the way we are working with these three different cell lines is applicable also to other cells. We don't need to use these cell types. We can also work with primary cells. This works as well. So why we're going into this complexity? Because when we look at the liver, this is a depiction or a schematic depiction of the liver. We see in gray here the hepatocytes, parenchymal cells, and they work in close interaction with you see these cells work with one that represents all the interactions between the cells. We need to incorporate more than only the hepatocyte. We can look at the liver, the organization of the liver, plate of hepatocyte. When we speak about the three-dimensional model, we are not representing exactly this structure. This is something that today we cannot do yet. We will have a multicellular model in 3D, in 3 dimensions as a spheroid, but not in this exact distribution. And why do we do that? Because we believe, and many people believe, of course, many scientists believe, that you can recreate the function by recreating the structure. So when you look at the HepaRG cells in 2D, you can recreate some functions, but obviously a two-dimensional flat culture is very different than the structure that a liver has. So therefore, we move from two-dimensional monolayer cultures to three-dimensional cultures. And monocultures with only one cell type to multicellular models, in this case with 3 different cell types. This didn't matter. So how can we generate these 3-dimensional models? There are several, oops, I think this went, sorry about that. There are several ways. This is not an exhaustive list, but this is the ways or the models which are used more commonly. On the one hand side, you have models which use a hanging drop approach, like the Insfero depicted here to the left top. And you see here in a 96-well format, these little drops inside or a spheroid, microtissue that is composed of the 3 different cell types. We can also generate agarose molds and you see here we have also very small wells, and inside each is made of agarose. We can generate a microtissue, and for today more important, the ULA plates, ultra low adherence plates. These ultra low adherence plates, you can study more this in a minute how this works. You generate the cell suspension and you put this cell suspension in this ULA plate. The cells will then go to the bottom, but they will not be able to attach to the plastic, and they will self-assemble into a little ball, and they will generate a single spheroid of microtissue. And we will see how this works in a minute. How can you do this practically in a minute? So the principle of this ULA plate. First, the ULA plate, as you will see also in the video, is a 96-well plate. It's in a 96-well format, and each of these wells, of these 96 wells, is a round bottom well. It's not flat, like we use normally for monolayer cultures, but it's a round bottom well. You put your cell suspension in your round bottom well in each of these wells, and the trick is that the ULA plates are coated specifically with an hydrogel, which is usually proprietary, which is covalently bound, hydrophobic, and neutrally charged, and repels the cells. So the round form of the bottom plus the coating hinders the cells of attaching to the plastic. The cells can then not attach. Normally, when you put cells into a plastic or into intracellular matrix, what you did also in your 2D cultures, you put the cell is not adherent because it comes in a suspension, but as soon as you put it on this flat surface, the cell will start to attach and generate a monolayer. By having this round bottom and by having this hydrogel coating, we hinder the cell of doing that. And since the cells, the HepaRGs, and the other cells we are using tend to be adherent cells, they will adhere to each other, and they will aggregate into a spheroid or microtissue, which is, at the end of the day, a ball of cells. So that is the principle of how the cells aggregate, and you will see in a minute how this is performed. Now, a couple of words or a couple of slides on why do we want to actually do this. Why do we need to go in a 3D system? Why do we need to use several cell types? And this is because we want to use the cells for complex models. Now here is a picture of how this looks like. HepaRGs, THP1, hepatic stellate cells, immortalized, all together they form one microtissue. And what I forgot to say earlier, the microtissues have a diameter of microtissues which are larger than approximately 300 micrometers. They become necrotic. The nutrients and the oxygen cannot penetrate if the microtissue is too big, and the cells in the core start dying. But our microtissues look like this. You see the different cell types with different colors in this depiction, starting from a monolayer. And as I was saying, we need to use this complex structure because for many liver diseases, we need intercellular interplay. So we need more than one cell type. We focus our research mainly on fibrosis, also cholestasis, and also NASH, the accumulation of lipids. And you can see in these very simple schemes that these processes, starting with the human liver, if you have accumulation of bile acids, that might lead to liver fibrosis and liver cirrhosis. Similarly, accumulation of fat might lead to liver fibrosis and cirrhosis.
Laura,
so this, processes, in particular the processes in fibrosis, yes?
Sorry, there's a
Yes?
connection issue, I think, because we can't really hear you properly. It seems to be cutting out.
Oh.
Maybe you should stop your video, Laura.
Yeah, maybe it would help.
To help the sound to. If you could turn off your video, Laura. Yeah.
I did.
Yeah.
Okay.
Okay.
Yeah, maybe it will help now.
Excuse me, I didn't, sorry about that. Can you hear me better now?
It seems to be a little bit better, so maybe we could try like this.
Okay, let's hope, huh?
Yep.
Okay, thank you. But okay, what I was saying is for the diseases that we try to investigate, that we're interested in my group and in many other groups, we need sometimes the interplay of the different cell types. In particular, the development of fibrosis involves not only the hepatocytes we can replace with the HepaRG cells, but also involve the Kupffer cells, which we replace with THP1s, and in particular the stellate cells. So that's why we use these models. The last part of this short presentation is only a few slides now, is how can we determine the response of a model? Once we generated these microtissues, we need to be able to analyze them. Can you hear me a bit better?
Yes,
Yeah, it's a lot better now.
Thank you. So the endpoint, there are many endpoints that one can imagine, but some of them which will be discussed or at least we use regularly are, for example, the cell viability. We need to know whether our microtissues are suffering, the cells are dying, and for that we measure ATP content. This is highlighted in blue because it will be shown practically as well in a minute. Then we can do PCR, so we can lyse the cells in a similar manner as the lysis that we need to do with the ATP, but with a different buffer. And we can then analyze the gene expression of specific genes inside these cells, inside these microtissues. We can also look at the deposition of the extracellular matrix, so ECM stands for extracellular matrix, or for other cell-specific markers using immunofluorescence, and you will see some of this as well. We can also look at the deposition of extracellular matrix or other specific markers using common histology, cutting the microtissues and doing histological staining. And of course, we can take the medium, collect the medium from these microtissues and analyze which proteins may have been secreted using a normal ELISA assay. We will not show you that. We have, however, also used the microtissues to do single cell RNA sequencing, which I will just show a set of data very briefly, but we will not show you practically because it's actually rather complicated. And of course, you can add any other endpoint that you may feel is important for your research, metabolism, for example, or other things. So in the video that you will see in a minute, not only will you see how to generate these microtissues and to handle them, but also how to assess two characteristic endpoints, the cell viability assay and the deposition of extracellular matrix. And I can show you very briefly is how actually this data that you can generate with these microtissues may look like. Again, I don't have the time to go into a big lecture on the toxicology of bile salts. It's not my intention. I just wanted to show which type of data you may be able to obtain using these methods. On the one hand side, I mentioned ATP assay for viability. This you're gonna see in a minute. There are other assays for viability. Here we compare two different assays. You see that the bile salts are actually toxic. to the cultures, and you can determine the toxicity and the IC50 as you may be used to do. We can analyze gene expression. As I mentioned before, you can lyse the cells, extract the RNA, and measure the expression of specific markers. In this case here, cytokine for inflammation markers, and here extracellular matrix protein fibrosis markers. And we can see that we have an increase in this particular case. But these kind of data are easily obtained once you have the microtissues and you have exposed them, treated them with a specific substance. In this case, bile salts at different concentrations. We can also do imaging. We can use immunostaining, either with immunofluorescence. You will see how this is performed in a minute. So we can stain here. We see these are 3D microtissues, so spheric microtissues. We see here the staining of the cell nuclei, and here the staining, for example, of fibronectin, one of the extracellular matrix proteins, which is induced by TGF-beta. So this is the whole microtissue. We're staining here a whole sphere, and we need specific imaging tools to look at them. We can also fix these microtissues, cut them, and make histology slides. And you can see this is just a slice, a thin, flat slice, a cut through the middle of this microtissue. And we can also assess the expression, for example, of collagen or alpha SMA or specific markers that we are interested in. We can quantify the amount of stained marked proteins there. So these are examples of the type of data, and if you're more interested in the background, here is the publication where this data came from our lab. I mentioned also before, we can collect the medium and see what the microtissues have secreted into the supernatant. In this case, we looked at interleukin-6 as also an inflammation marker, and we can see that TGF-beta and also bile salts induce the secretion of IL-6 into the medium. And last but not least, and this is something that you will not be able to infer from the video that we're going to show with the practical part. We have it is possible, but relatively technically challenging, to generate data using single cell sequencing. So once we have assembled the microtissue, this is a representation of the microtissue, you can keep the control on treated microtissues, and then it's a relatively complex process of dissociation, so generating again a single cell suspension, of marking the single cells, and then generating the sequencing, the single cell sequencing data and analyzing this data. This is absolutely out of the scope of this demonstration, but it's one possibility that I wanted to mention to you, because then it allows to see, this is the data from single cell sequencing, which is, as I said, not discussed in the demo, but we put 3 cell types together, HepaRG, stellate cells, and THP1s, and we can see, based on the gene expression of each of the cells, each dot here is a cell, it's not anymore a microtissue, it's just a representation of the gene expression of a cell. We can see that we have the pink colored cells are the HepaRGs, the green ones are the hepatic stellate cells, and the blue ones, the macrophages. So we are able to separate them based on the gene expression, and we can also assess the response. You can see, for example, that in TGF-beta treated microtissues, we seem to have an abundance of THP1 cells, which is something expected. We will not discuss this in the demo though, but it's a possibility. So using a 3-dimensional multicellular system is many different things that you can actually address. So this is also all the different things are published in several publications from my group. So if anyone is interested, and we can do it later as a follow-up into the different topics. But now we would really like to focus more on the practical approach, and this is what I'm going to try to do now. I'm going to try to get you to the virtual lab.
Today we will show you how to generate 3D liver microtissues using ultra-low adherence plates. Before starting, you should prepare all the material you will need. PBS, trypsin and aggregation medium should be warmed at room temperature. The aggregation medium contains Williams E medium, Glutamax, ITS, 100 nanomolar dexamethasone, 20% FBS and 1% penstrep. You will need to prepare trypan blue and a Neubauer chamber for cell counting. And this protocol then involves ultra-low adherence plates, also known as Euler plates, which we will describe in more detail. However, they facilitate the formation of spheroids in a scaffold-free manner. There are other methods available, such as using the Sigma micromold system or the InSphero hanging drop technology. However, in this case, we will only be showing you the method using Euler plates. Today, we will generate 3D human liver microtissues using 3 cell lines, so this is a triple model. We will use the cell line HTERT-HSC, which are an immortalized human stellate cell, and was kindly provided to us by Dr. Bernd Schnabel. We will use the human monocytic cell line THP1 from Cell Line Services, and these are differentiated into macrophages using PMA for 48 hours, followed by a 24-hour rest period prior to use. These are a surrogate for Kupffer cells. Finally, we use the human cell line HepaRG from Biopredict International, and these HepaRG are differentiated in a specific differentiation medium for 2 weeks prior to use, and they serve as a great surrogate for hepatocytes. Cells should be observed prior to starting, so check for confluency and record passage number. Passage numbers are important for our cells, as THP1 can only be used up to passage 30. The HTERT-HSCs can be used up to passage 12 and the HepaRG up to passage 20. We also check microscopically the differentiation status of the HepaRG and the THP1 cells. And of course, we check for spontaneous activation of stellate cells to ensure the cells are in the appropriate condition prior to starting. Now we will proceed to the generation of microtissues. All steps will be performed under a sterile cell culture hood. Ensure the hood is clean by spraying it with 70% ethanol. Bring all of the items that you need, so PBS, medium and trypsin, and spray them with ethanol prior to transferring them under the hood. You will also need to bring your flask of cells. These do not need to be sprayed with ethanol prior to bringing them into the hood. We will start with the THP1 cell line, since these require the longest trypsinization step, so it takes approximately 20 minutes. However, the same procedure is used for the 3 cell types. Initially, medium is carefully aspirated. Then the cells will be washed with 10 milliliters of PBS. Following this, the PBS will be aspirated and we will add 2 milliliters of trypsin to the flask. The trypsin will be evenly distributed across the flask, and then the flask will be brought to the incubator, which is at 37 degrees centigrade with 5% CO2, and there the 20-minute incubation will begin. The same trypsinization procedure will be performed for the HepaRG and the stellate cells. The HepaRG will be trypsinized second, as they are the second fastest to detach from the flask, and then the stellate cells will be trypsinized last, as these detach the quickest. Following the incubation at 37 degrees, tap the flask to ensure that the cells have fully detached. To confirm that trypsinization has worked and the cells are detached, these can always be checked at the microscope under a bright field setting. Here we see the HepaRG after trypsinization as an example of this. As you can see, the HepaRG will need to be mixed well prior to use to ensure a single cell suspension. The cells are then resuspended in aggregation medium, which contains 20% FBS and therefore inactivates the trypsin. Aggregation medium is added to the flask and the cells are mixed. The amount of medium added is dependent on the density of the cells within the flask. For example, stellate cells are quite large and are not generally dense in the flask, therefore require only a total of 5 ml before counting. Whereas HepaRG are smaller and there are many more cells in the flask, meaning that counting using 5 ml would be very difficult, so we resuspend in larger amounts of liquid to have a final volume of 10 ml. Once the cell suspension has been collected, it is transferred to a sterile 15 ml falcon so that the cells can be stored and used later. The same procedure is performed for both the HepaRG and the stellate cells. Now that all cell suspensions are prepared, 20 microliters of each one will be pipetted into 3 different 1.5 milliliter Eppendorf tubes. For counting, trypan blue exclusion method is used. This is based on the principle that live cells possess intact cell membranes that exclude certain dyes, such as trypan blue, whereas dead cells do not. Therefore, by excluding dead cells, we will obtain a viable cell count. Trypan blue is added at an equal volume to the cell suspension, so in our case, 20 microliters. 10 microliters of the mixture is then loaded into the Neubauer chamber to allow counting of total viable cells. The Neubauer chamber remains the most common method used for manually counting cells. Counting of viable cells is performed under a light microscope following the procedure of the Neubauer chamber. All 3 cell suspensions are counted and the desired cell numbers are calculated. It is important to determine beforehand the number of microtissues you will need to calculate the volumes of each cell suspension to pipette. Today, we are preparing 1 plate of 60 wells with the microtissues in. The microtissues are made up of 2,000 cells at a ratio of 2 to 1 to 1 of HepaRG to stellate cells to THP1. This information has been published and will be provided to you at the end of the presentation. As we are using 60 wells, we will therefore have 60 microtissues. However, all calculations are made including an excess of 10 microtissues, so in this case 70 microtissues. This means that a total of 140,000 cells are required. This will ensure that enough cell suspension is made to create the desired number of microtissues. The desired volume of each cell type is obtained by dividing the required cell number, for example in this case 70,000 HepaRG, by the calculated cell density. In our case, based on our cell counts, we are now pipetting 107 microliters of HepaRG with 120 microliters of stellate cells and 270 microliters of THP1 cells. This cell mixture is then brought to a final volume of 14 milliliters. This is calculated by using the number of microtissues, in this case 70, by the volume required per well, in this case 200 microliters. The protocol of microtissue generation involves an ultra-low adherence plate, or a Euler plate, as already mentioned in the beginning of the video. These plates are round-bottomed, clear 96-well polystyrene microplates. The ULA coating, which is hydrophobic, in the round-bottom wells facilitates the formation of round spheroids after seeding. The cell suspension containing the 3 cell types is now transferred to a reservoir to allow pipetting with a multi-channel pipette. 200 microliters of cell suspension are pipetted to each well of the Euler plate. No cell suspension is pipetted into the outer wells, as here we have the risks of medium evaporation and malformation of the 3D microtissues. To avoid evaporation within the plates, we pipette 200 microliters of PBS into the outer wells. Proper seeding is checked under a light microscope. At this stage, cells are still dispersed in the wells. The formation of the 3D spherical microtissues will require 3 days of cell aggregation at 37 degrees in an incubator. Following the 3 days of aggregation, the formation of the microtissues is checked under a light microscope. Here we can see the formed microtissue. The size of the microtissue is approximately 250 micrometers. A medium change to an FBS-free version of the medium, or treatment of the microtissues with different substances, can now be performed every 2 to 3 days until endpoint. The microtissues are now ready to be analysed. We will now show you how to perform an ATP viability assay, and the work can now be performed under non-sterile conditions. We are using the Cell Titer Glow Luminescent Cell Viability Assay from Promega. It is a homogeneous method to determine the number of viable cells in culture based on quantitation of the ATP present, which signals the presence of metabolically active cells. The homogeneous assay procedure involves adding a single reagent, the Cell Titer Glow reagent, directly to the cells. By adding the ATP reagent into the wells, it results in a cell lysis and the generation of a luminescent signal proportional to the amount of ATP present. The amount of ATP is directly proportional to the number of cells present in the culture. For the cell titer glow reagent, organize an orbital shaker to perform cell lysis, a multi-channel pipette, a white opaque plate to measure the luminescence, and have an appropriate plate reader. Carefully remove 100 microliters of the 200 microliter medium in the well. This step is required to avoid loss of microtissues that may happen with a complete medium removal. An equal volume of cell titer glow to medium is added, in this case 100 microliters, and the cells are then lysed on an orbital shaker for 10 minutes at 500 RPM. The lysate is transferred to an opaque plate to prevent the wells from interfering with one another. The luminescence is then measured on an appropriate plate reader. Background luminescence is removed by measuring a well that contains no cells. 100% of viability is set to the control, i.e. untreated wells, if microtissues were subjected to a drug treatment. Here we show an example of a drug treatment that was performed. So spheroids were exposed to paracetamol, and we can see that by measuring the ATP and comparing it to an untreated control, that we have a relative decrease in ATP. We will now show you how to perform a staining procedure and imaging of the 3D human liver microtissues. This work is done in non-sterile conditions. We will first proceed to the fixation of the microtissues, which will be carried out under a chemical fume hood, and then we will proceed to the staining procedure, which can be performed at a normal workbench. Under a chemical fume hood, as paraformaldehyde is toxic, prepare 8% paraformaldehyde, so PFA, a 1x unsterile PBS containing calcium and magnesium, and bring a reservoir and a multi-channel pipette. Then, when ready, bring your Euler plate with the 3D microtissues. 150 microliters of medium is removed from the wells with a multi-channel pipette. Again, complete medium removal could lead to loss of microtissues. 150 microliters of PBS supplemented with calcium and magnesium are added to the wells, and this is repeated once, and they are then incubated in this PBS for 10 minutes. This step, using calcium and magnesium, avoids disassembly of the microtissues, as it ensures cell adherence. Following this, 150 microliters of the medium/PBS mixture are removed, and the microtissues are fixed with 50 microliters of 8% PFA. As 50 microliters of PBS is left in the well, the addition of this 8% PFA will result in a final concentration of 4% PFA. Following the 1 hour fixation period, PFA is progressively removed and replaced with PBS with multiple washes. Now we will show you a staining procedure using phalloidin. For this procedure, we need a 1x PBS. We need a permeabilization buffer, which is made using 1% Triton X100 in PBS. We will use Alexa 488 phalloidin as a phalloidin conjugate used to stain actin filaments in fluorescence microscopy, and DAPI as a nuclear marker. We are using black imaging 96 well plates with a clear glass bottom. The glass bottom plates offer excellent planarity for sophisticated microscopic analysis. Additionally, black plates offer an excellent signal-to-noise ratio. A rocker is also required for this process. Following the fixation procedure, which we have just shown, the microtissues are washed with PBS. Microtissues are then permeabilized for 4 hours at room temperature on a normal workbench, so again in non-sterile conditions. "50 microliters of the PBS is removed and replaced by 50 microliters of permeabilization buffer. This will obtain a final 0.5% Triton solution, which is sufficient for permeabilization. Following the permeabilization, remove as much permeabilization buffer as possible and proceed to wash with PBS 3 times. As mentioned before, today we are using Alexa 488 phalloidin, in which a blocking step is not required. However, when using a primary antibody combined with secondary antibody, a blocking step is required, and typically we will use 3% BSA and also block for 4 hours or overnight at 4 degrees C. The mixture of Alexa 488 phalloidin and DAPI are prepared at the appropriate dilutions in 1x PBS. These can then be pipetted into the wells. Following an hour on the rocker, we can remove the phalloidin DAPI mixture and proceed to PBS washers. Once the staining is complete, we can transfer the stained microtissues to a black imaging plate using a multi-channel pipette. 1 microtissue is transferred to each well. However, some microtissues may get lost at this stage, so it is always important to have enough replicas of each well just in case. Your plate is now ready for imaging. In order to image 3D structures such as microtissues, laser scanning using a confocal microscope is the best method. Using confocal microscopy, the sample is illuminated with a point source of light, and we are able to take sectional images of the sample through the entire depth of it. Reconstruction of the 3D structure is operated by stacking these slices back together. The staining of the very internal part of the microtissue can be seen with better clarity using a confocal microscope. However, it is still not possible to image directly through to the core of the microtissues. Here we show the resulting phalloidin staining at the confocal microscope, and we are showing how the imaging is performed, and you can see this progressive reconstruction of the 3D structure. This, of course, is done for both channels, the phalloidin and the DAPI. So at the end, we can create a merged picture. Here we see the final image of the microtissue stained with phalloidin. We have also provided some additional images of staining that requires the primary and secondary antibodies as examples of the other process that can be performed.